Well, good morning, everyone. I just need to adjust this to Dutch levels. I'm absolutely thrilled and particularly honored to be invited to be the speaker for this morning for the Kees, the Lange Memorial Lecture. And of course, John Patience gave the introductory lecture last year, and together with Meng Fang here on faculty here, they wrote a very nice biography paper that was published earlier this month with page numbers in the American Journal of Animal Science. So if you would like, some of you may not have access to a library. If you would like to have a copy of this paper, please contact one of the organizers or myself. Like my email, I believe, is in the proceedings or on the final slide. And so John did a very nice job at last year's lecture talking about Kees and his accomplishment. So I will not reiterate all his accomplishment one more time, but I will focus a little bit of Kees's his origins. And of course, his origins are very similar to mine, is that he was born and raised in the Netherlands in a tiny town called De Veere. So what about the Netherlands and what about Europe? Well, I'm sure you didn't come here this morning to learn about the Netherlands and that it's actually located here in Western Europe, between Germany, France, UK, as some other neighboring countries. But then when you look at the country itself, you know, I kind of eyeballed it out, that if you, just to give you some idea of the geography, if you go from Amsterdam, which is the capital, to the place where my folks live in Zwolle, that's the same distance as from here to downtown Toronto, right? So this is the size of the country. So with a good car, 2 hours at the most east-west, a very good car, 3 hours north-south. But that country is the second in the world when it comes to agricultural exports in value, next after the US. So question that I have for you is, what do you know about Dutch people? Don't be afraid to raise your hand. One I'll start with, they tend to be tall, right? Dutch, you know, the original nationality, and indeed the name, de Lange, means the tall one. And when I go back home, I'm an average guy, right? So that's one. What is another one you would know about?
Very determined.
Very determined. Another way of saying that might be stubborn, right? So you can think of the saying that I used to like to use is wooden shoes, wooden mills, wouldn't listen, right? And what is another one that you may know of? Speed skating is one. Okay, and this determination is particularly strong in the northern part of the Netherlands, to that extent that 2,000 years ago, this was the border of the Roman Empire. They didn't want to deal with those guys in the north of the Netherlands. And the final one is, you may know about, is getting things done on time. Okay? And what we tell our kids is, there's no such thing as being too early. You're either on time or you're late. And then when you then focus in this area of the Netherlands, North Holland, that's where Kees was from. Particularly this area here, West Friesland. So Kees's hometown is right here. And what you see here is basically the old dike system that was developed around West Friesland. And if you go back, this is all land that was reclaimed right before the Second World War. This land was reclaimed in like the late 1800s, and there's pockets of land that were reclaimed here in like the 1600s and the 1700s. And if you then go back 500 years ago, that same plot of land, this is how it looked like. This was all sea. And this diking system was developed in around 1250. So my theory of why Dutch people are on time is that before this diking system was developed, you were either on time on the high ground or you would drown. So I think it's the first case of genetic selection, if you will. And there might be some other reasons why that might be, but that's my theory. And of course, I'm sticking to it because I'm Dutch. So then in our nutrition program, in our lab, we basically focus on 3 areas. The first one is looking at the impact of dietary carbohydrates. So this is starch, fiber, oligosaccharides, sugars, etc., on digestive physiology. And I'll focus on that today. Second one is to look at all kinds of different feedstuffs, grains, cereal grains, co-products that we produce in Canada. And it's actually quite an impressive array of co-products that are being produced, and characterize their nutritional role and functional role. And then the third one is to look at rapid feed evaluation. Now, all of these, they actually work in concert as well, and this was certainly one of Kees's pet peeves, is make the pigs less dependent on foodstuffs that people can eat, and make them more dependent, let them eat more co-products that people do not want to eat. I'm sure you didn't have any canola meal for breakfast this morning, but the pigs can convert canola meal back into the sausage that you might have had for breakfast this morning. I certainly had one this morning. So when you then look at starch and fiber as carbohydrates and how they originate, well, they're all plant-based nutrients. And in the plants, you have the cell walls. Plants don't have a skeleton, so they use a very rigid cell wall to stand up. And that's where you have molecules such as lignin and cellulose that in animal nutrition we characterize it as a detergent fiber. Then we can also add the hemicellulose, and then you will have a neutral detergent fiber. And both these two fiber analyses, they characterize insoluble fiber. And then in human nutrition, we also use total dietary fiber, where we also bring in the soluble fiber component and analyze it together as a TDF. And then in cell content, you can have like organic acids, sugars, fructans, but of course in cereal grains, that's where you find the starch. There's a lot of starch in the cell content of cereal grains. But this distinction is not exactly true when it comes to functionality. So any starch that is not digested can potentially become fermented in the gut, and then it basically acts like a fermentable fiber. And using that functionality allows us to study the role that the different kinds of starches may play within the pig gut. One of the reasons why we use this information is try to provide solutions for post-weaning diarrhea. So this is still an issue in modern swine production. PED was a more recent case, but also by removing antibiotics from swine diets, the chances of pigs developing diarrhea soon after weaning goes up. And there's a couple of components of why this is happening. So immediately after weaning, there can be a reduction in feed intake of these pigs, and that in itself will immediately cause the gut wall to deteriorate. Because the gut wall is deteriorating, there might be less functionality when it comes to digestive power within the gastrointestinal tract. You can end up with more undigested residue, and if that undigested residue starts to ferment, particularly when there's protein there, you get protein fermentation. And this is something very bad to happen in the pig gut. Because of this gut wall integrity, protein fermentation, you can have more chances of inflammation because you can end up with an unstable microflora, etc. So when these antibiotics are not available anymore, or not used anymore to control gut health, we need to find alternate solutions. So we can think of organic acids that are being used, probiotics or live good bacteria that are being used, and another tool is the prebiotics. And these are selectively fermented ingredients. That's one key component of their definition that causes specific changes in composition of the microbiome, functionality of the microbiome, and as a result provides a benefit to the animal. So most of these prebiotics, they're fermentable carbohydrates. So short-chain fibers, oligosaccharides fit that category, resistant starch, so the starch that is not digested but fermented, and also pectin from sugar beet pulp, beta-glucans that can come from cereal grains, can all be prebiotics. And then, of course, the question is, for me as a nutritionist, am I going to use the original feedstuffs to provide prebiotic activity, or am I going to fractionate out and purify, for example, inulin from sugar beet pulp and provide the prebiotic as a feed additive? Anytime when you talk about prebiotics, there's a flip side to that coin, and that is energetic efficiency in the pig. So I have two scenarios for you here. The one on the left is a diet that has a low amount of resistant starch or a low amount of fermentable fiber. And that means immediately you have a high amount of digested starch. That gets converted into glucose, and you have very little starch and fiber that is converted by the microbes into short-chain fatty acids. And then the scenario on the right, now we have a diet that is high in resistant or fermentable starch and fermentable fiber, so you have less digestible starch that is converted into glucose, and then more fiber and starch converted into short-chain fatty acids. But there is an energy cost when this happens, right? Nutritionally, we call this the heat increment, and it basically means that with fermentation, there are greater energy losses. Now, how do we then make use of that looking at starch? Well, starch is just not one molecule, there's actually two molecules that make up starch. One molecule is amylopectin, which is organized like a helix, so think of like DNA. So this is a very rigid starch molecule, very difficult to digest by pancreatic amylase. And in contrast to that, you have amylopectin, which is a branched starch molecule, so very open starch molecule, very quickly digested by pancreatic alpha amylase. So we can develop, and that's not me, that's the crop breeders, cereal grains with different ratios of amylose to amylopectin. So a normal cereal grain, you would have about 10% amylose and 90% amylopectin. But using crop breeding, we can end up with starches that are either 100% amylopectin, so a very rapidly digestible starch, waxy starch, or starch that can be up to 63% amylose, so 37% amylopectin, so very slowly digestible starch. And then using wet fractionation, we can lift that starch from the cereal grain and now have purified starches that can range drastically in their starch chemistry. And then, of course, as nutritionists, we can make use of that. Now we have purified starches by keeping the rest of the diet constant, have the same amount of digestible protein sources, fiber, energy, vitamins, and minerals, and then by changing the starch source in the diet, drastically change how the starch is digested within the diet as a whole. Then, of course, we use, these are pigs, so we use sophisticated models to see what's happening. So one of those models, also used by Kees here, was the ileocannulated pig model, where we install a cannula in the distal small intestine, and when that cannula is closed, we have normal digestive flow, we collect feces, and can determine apparent total tract digestibility of energy, starch, or any other nutrient. Then we can open the cannula. Now we collect intestinal digesta and can determine ileal digestibility of those same components. And then the other thing we can do is then by difference determine how much of energy of a nutrient has been fermented in the large intestine. So we did that for those 4 starches. So the first panel on the left here is the amount of starch that is left intact at the end of the small intestine for those 4 starches. And these slides are color-coded. I hope you're not colorblind, right? But I organized the red very far away from the blue. So rapidly digestible starch, so the low amylose starch is in red, and then the blue is the slowly digestible starch or high amylose starch. So what you can see here is that when starch is very rapidly digested, there's no intact starch left at the end of the small intestine. It's all been digested. And then for the third starch, there's about 3% of the starch is left intact. And then of the slow digestible starch, about 30% of the starch is still intact at the end of the small intestine. Then with these purified starches, when we then look at the feces for starch, it's basically all gone, meaning that any of those purified starches that entered the large intestine was completely fermented. And this is actually potential benefits, when you think of pet food nutrition, for example, complete fermentation of these prebiotics within the gut is a good thing. You can imagine that you end up with very different glucose and short-chain fatty acid fluxes in the gut, causing all kinds of changes in the gut wall. I don't have time to talk about that this morning, but you can think of different transport mechanisms, gut integrity molecules are all being affected by the changes in nutrient flows. Another model that we can use is the portal vein catheterized pig. So here you see the heart system, here you see the GI tract and the liver, the lungs. And what we do in this model, in the portal vein, which is the blood vessel that drains the gastrointestinal tract, we install a catheter right upstream from the liver, and then also install an ultrasonic blood flow probe to measure blood flow through this portal vein. Then we install a catheter in the carotid artery that basically reflects blood flowing towards the gut. And then we can, following a meal, we can sequentially analyze blood samples or plasma samples from both these, collected from both these catheters, and then end up with net nutrient absorption and basically add a kinetic component following a meal to nutrient digestion. The interesting thing about this model is that the pancreas also drains into the portal vein, so insulin is drained there, and then the gut itself also makes hormones. We call those incretins that are also drained into this portal vein. So by looking at this model, we can look at how nutrients flow in and how the body is responding with its endocrine system. So using those four same starches again with portal vein catheterized pigs, if you first look at the top panel, here we're looking at net portal appearance of glucose following a meal for those four starches. So that rapidly digestible starch, you see a tremendous peak. You also see a very large area under the curve. So this starch, now in human nutrition lingo, has a very high glycemic index. So that's a starch you want to avoid when you have type 2 diabetes. Right? And then in contrast to that, you have the slowly digestible starch. You see that the peak is reduced by about 50%, and you see a prolonged and a more stable uptake of glucose from the gut. Then at the bottom, we look at now at net portal appearance of insulin, and you can see that the things that I learned in school, and hopefully you learned in school here at Guelph, is that when you have more of a glucose response, you have more of an insulin response as well. In other words, high glycemic grain or starch gives a bit greater insulin response. But there's something else in this slide, and that is the timing. And when you just go down from the peak of glucose, it's actually happening after the peak of insulin. And this certainly gives us reason to believe there must be some signaling happening from the gut to the pancreas to get ready and secrete insulin, because there's a massive load of glucose coming down the portal vein very soon. And that's perhaps where these incretins can come into play. So incretins are hormones that are produced by the gut themselves. There's about a dozen of these, and two of them I'll introduce this morning. The first one, glucose-dependent insulinotropic peptides, for obvious reasons abbreviated as GIP. And this gut hormone is being produced by enteroendocrine cells, so hormone-producing cells within the gut that are called Kees cells that are located in the first and the middle part of the small intestine. Okay? And these Kees cells, they release GIP when they sense that there's glucose in the lumen of the gut. And then in contrast to that, you have another incretin called glucagon-like peptide 1, which is being released by L cells, which are more in the end of the small intestine, the cecum and the colon, and they release GLP-1 when they sense that short-chain fatty acids, so the mediators that are produced by the bacteria, are being released in the lumen of the gut. So using that same portal vein catheterized pig model, we can look at areas under the curve in the same kind of time sequences. These hormones are released in a pulsatile manner, so we decided to look at particular time periods following feeding for those two different hormones. So 2 to 4 hours after feeding, the digestive is now flowing through the small intestine, and indeed, when this rapidly digestible starch is quickly digested into glucose, we see the most amount of GIP being taken up in the portal vein, and the lowest amount for the resistant starch. And then in contrast to that, 4 to 10 hours after feeding, now the digestive is flowing through the end of the small intestine and the large intestine, we see the opposite for GLP-1, right? So now the starch, whatever starch is still there, is being fermented, and now this GLP-1 is being released. Now, some of you may have pigs that are housed in group housing, like, for example, in sows, and when you feed, for example, more fermentable fiber to sows, you have a calming effect on the animals. Well, this is actually the mediator hormone that makes that happen, the link between fermentable fiber to changing in behavior using this mediator. We can, of course, also look at short-chain fatty acids. So first, in the cannulated pig model, we can look at concentration of short-chain fatty acids in feces and digesta. And indeed, when you have more fermentable starch, you end up with greater concentration of total short-chain fatty acids in fecal material. But even already in the ileal digesta, you can find more short-chain fatty acids, indicating that starch can be fermented at the end of the small intestine. And that, of course, is in line with the work that Kees did, looking at amino acids, that indeed microbial protein can already be formed in the small intestine and make like a contribution of 5 to 10% of meeting the requirement for essential amino acids in the pig. Now, of the short-chain fatty acid, butyrate is a very important one because it provides energy to the colonocyte, so the cell lining in the large intestine, and it's also a very important signaling molecule within the gut. You can see that when you have more resistant starch, you produce more butyrate. But we can also look at this data using the portal vein catheterized pig model, because short-chain fatty acids are absorbed into the blood, unlike large-chain fatty acids that are absorbed into the lymph. So what's interesting here is that net portal appearance of total short-chain fatty acids is exactly the same pattern as concentration in fecal material, but for butyrate, that difference is greater. So what we think that is happening here is that when you have a certain amount of butyrate that is being produced, all of that is being used as an energy source by the colonocyte in the large intestine. But when too much butyrate is being produced, excess butyrate will leach into the body and will be part of ketone body energy metabolism. Short-chain fatty acids, of course, are produced by microbes, so you might expect that when you provide changes in fermentable substrate, you would see massive changes in the microbiome. And that's actually something unique for resistant starch, because it's not happening there. Right? So here, when we look at gene copies in grams of feces for total bacteria, some Clostridia clusters, Enterobacteria, say Bacteroides, absolutely no change in density of these bacteria, and that's the first indicator that bacteria diversity wasn't affected. There's a little bit of a trend that resistant starch might increase lactobacilli. However, there's always one classical effect of resistant starch, and that you see an increase of bifidobacteria. And we call this the bifidogenic effect of resistant starch. Now, bifidobacteria certainly have been associated in many models with having a better gut health, but it's not a causal effect. In other words, we can see there's more bifidobacteria, so gut health might be better, but they're actually, what we think, we do not fully understand how they actually contribute to better gut health. The other thing that it of course means is you can have massive changes in fluxes of short-chain fatty acids with minor changes in bacteria profile, indicating that the bacteria pool overall can become much more productive in producing short-chain fatty acids. All the data so far I did with grower pigs, but there is some evidence that this can happen in nursery pigs as well. I'm not sure what this panel is missing. So when you look at in nursery pigs, we see the same bifidogenic effect in the small intestine of nursery pigs. We see the same amount of short-chain fatty acid increase as what we see in nursery pigs as in grower pigs, and we see some minor changes in the microbiome. Well, Mr. Swine Professor, this is all very nice, but what does it now really mean for my pigs? And that is a link that is still missing. Do these changes in the microbiome, these changes in short-chain fatty acids that are being produced, really matter when it comes to gut health? And the data on there is actually very sketchy and very poor in quantity. And one of the studies that I have here for you is from Dennis Krause, who used to be on faculty at University of Manitoba before he passed away, together with Martin Iaciotti, where they looked at two models where they looked at raw potato starch. So this is not really fermentable resistant starch, but it's somewhat fermentable. And what they did in their model is first diet had a positive control with an antibiotic, then removed the antibiotic, and then 2 incremental levels of raw potato starch. So by removing the antibiotics, the pigs did worse, and then by adding more raw potato starch, they certainly did not improve their performance. But when you look at fecal score or fecal consistency as an indicator of gut health, when you remove the antibiotic, fecal consistency certainly became a little bit worse. By adding raw potato starch, it improved at the low dose, but not at the high dose. No mechanism was studied whatsoever in this study. And then a second study where they had an E. coli challenge, also between the positive control and the raw potato starch, not much of a difference between the two treatments. So my statement, one of my statements this morning, that impact on gut health is, needs to be studied relative to resistant starch and carbohydrates. Of course, energetic efficiency can be studied in other models, and what you see here is digestible energy is declining with an increase in starch fermentation. Well, this contradicts what I told you 10 minutes ago, that all the starch is digested and fermented within the pig gut. So how can this happen? Well, this resistant starch is basically an energy source for the microbes, and then the microbes will produce more microbial protein. That microbial protein is excreted in fecal material, and that's why total tract energy digestibility, the apparent and DE goes down. And then using indirect calorimetry, we can certainly see that the net energy value of increase in fermentable starch causes a reduction in net energy. When we then feed those starches to nursery pigs, okay, we can see that, and this is immediately after weaning, we can see that there is a tiny reduction in daily gain because more undigested material may start to limit feed intake. You certainly see a reduction at a high level of daily gain. And then when you look at feed efficiency, you can see like a 5 to 10% reduction at resistant starch. But we also see a curvilinear response here for rate of starch digestion. So that's enough about starch. So a few comments on fiber. So fiber, traditionally in swine nutrition, we separated as soluble and insoluble fiber, and then made statements around solubility describes viscosity and fermentability. And actually what we do now know using purified fibers, that's not the case. So viscosity is a unique effect of some fibers that basically has a role to play in how fast material is being released from the stomach into the small intestine. So for example, more viscous fiber in the diet can have a dampening effect on that net portal appearance of glucose that I described earlier. And then fermentability of the fiber describes how much of the fiber is now fermented into short-chain fatty acids. So a little bit on viscosity using purified fibers, either cellulose, carboxymethylcellulose, or low or high molecular weight beta-glucan. These two are non-fermentable, these two are fermentable. And what you can see here for carboxymethylcellulose, you may see it on some of the labels of food products that you have at home. It has a massive dampening effect on rate of passage. So it drastically changes how fast the digester is flowing from the stomach into the small intestine. In contrast to that, looking at fermentability, so this is using this catheterized pig model again, where we concentrated beta-glucans from barley and fed at 2 incremental levels purified beta-glucans to pigs. And here you can see that when you feed purified beta-glucans, you see this clear prebiotic effect of an increase in the amount of short-chain fatty acids that are being absorbed by the pig. Fermentable fiber has some other nice side effects. This is actually data from Kees' lab. And I talked about protein fermentation before. Well, protein fermentation produces amines, and there's a whole array of them. I have theramine here, spermidine, cadaverine, if you believe it, is another amine that can be produced in the pig gut and can have nasty effects on the gut wall. But these molecules contain nitrogen, so they can serve as a nitrogen source for the microbes in the gut. So when you feed more fermentable fiber, you feed more energy to the microbes. Those microbes now start looking for nitrogen, and that's what you see the effect here, that by feeding more fermentable fiber, we can reduce the amount of amines in the gut and thereby also improve gut health. I'd like to finish up with a few slides where we actually went back and started to look at whole grains again. So there's 5 cereal grains I will introduce to you. On the right side, we had a regular wheat. And when you have a regular wheat, you have about 6% ADF, about 12% amylose, and virtually no beta-glucan. Then we switched to a hulled barley. So now we have the hulls, so you see ADF go up to 9, but you see amylose still the same as wheat, but now you also see beta-glucans appearing. Okay? Now, then we had 3 samples of hulless barley. So now you remove the hull, so ADF goes back to the sample of wheat, and then moderate fermentable hulless barley had the same amylose and beta-glucan profile as the whole barley, and then by either, again using crop breeding, increasing amylose or beta-glucan, we can drastically increase the fermentability of the carbohydrates within the pig gut. So the first thing we did was characterize these grains in ileocannulated pigs. So by feeding those 5 grains, first look at total tract energy digestibility. You can see that when you go from wheat to barley, you see energy digestibility go down because there's a hole there. And when you remove that hole in grower pigs, energy digestibility is the same as wheat again. So hulless barley, from an energetic perspective, provides the same energy to pigs. Then when you look at this total, this large intestine fermentability of either energy, starch, and beta-glucan, now you really start to separate those 5 cereal grains. So you cannot see that effect here, but here you can really see it. Right? That from low to high fermentable grain, you basically go from 10 to 40% of the energy being fermented in the large intestine, a slightly less of a difference for starch, and a kind of a doubling for beta-glucans. But the part of nutrients flowing through the gut, they're actually flowing through the gut in a 3-dimensional structure. And so one of the technologies we use in feedstuff analysis is confocal laser scanning. So this technology is actually used for cancer diagnostics, because cancer cells have a different cell surface markers than healthy cells in the body. So we can use coloring agents that bind to those cell surface markers and make them light up underneath a microscope. Well, we can do the same with starch. We can have coloring agents that bind to starch, and now the starch granules are lighting up underneath a microscope when the starch is still in the diet. Then, of course, we can collect digester samples, and then in the low fermentable, so highly digestible starch, we see no intact starch left at the end of the small intestine. Moderate, we see a few, but then certainly this high fermentable, high amylose starch, we see those intact starch granules that now will flow into the large intestine and ferment and hopefully provide a better gut health. When we then look at energy digestibility for these nursery pigs, and we fed these pigs were weaned at 3 weeks of age, and 1 week after that, we started to feed these 5 different cereal grains for 5 weeks, first phase for 2 weeks, and then the second phase for 3 weeks. And when you then look at digestibility of energy in these nursery pigs, you see a slightly different pattern as what I described for the cannulated grower pigs. The first part is the same. So when you go from wheat to barley, you see the reduction in energy digestibility. And when you then switch to the first hulless barley, the moderate fermentable hulless barley, you see the same total tract energy digestibility in nursery pigs as what you had for wheat. But then when you switch to the highly fermentable material, you can see that these young pigs, certainly right soon after weaning, they had a lower amount of apparent total tract energy digestibility. Question is, of course, how do they respond when you feed this material? And then the interesting thing is that for the first 3 weeks of the trial, we could not detect any difference in feed intake, but then, of course, feed intake starts to creep up because these pigs are getting bigger, and then when you get pigs, 4 in week 5 of trial, now you start to see changes in the amount of feed that they can consume. What is actually very interesting, if you switch nursery pigs from a wheat-based diet to a barley-based diet and control the diet energy profile, these pigs fed barley-based diets are actually doing very, very well. Then when you switch to the first hulless barley, they can still consume that diet to the same as the wheat or the barley. But then when you go to the highly fermentable grains, they cannot keep up their feed intake and therefore their energy intake. And that's therefore we detected for the entire trial as well. Then when you look at average daily gain, no difference in the first 2 weeks. We could detect significant differences in week 3, but not in the other, the final 2 weeks of the trial, not in the overall experiment, but of course, some of you might be pig producers. You may not believe statistics, so I have a comment here at the bottom, right? That final body weight of the pigs at the end of the trial of this highly fermentable grain tended to be about 1 kilogram less than of the highly digestible cereal grain. So I like to finish up with this diagram and kind of link starch to health and growth. So if you would be a human, and you would be my age, and don't want to offend anybody, or be slightly older than me, I would recommend eat all the resistant starch that you can, because what does that now do? It will reduce the energetic efficiency of what you consume, and to make you less obese or make you lose weight. I'm not claiming that I had resistant starch this morning, just to be clear. But then this increase in starch flux into the large intestine in the human gut has all kinds of positive side effects when it comes to gut health. Right? Colon cancer risk goes down, etc. But of course, in pig production, we have some other considerations because we like our pigs to grow and reach market weight within a certain reasonable amount of time. So we may have come to a different conclusion and have a certain amount of resistant starch or prebiotic activity in the diet, but not too much. We still would like to see the positive effects on gut health, but not the negative effects or severe negative effects on body weight gain. So I like to conclude with these comments that dietary carbohydrates can have potentially a very strong prebiotic effect, particularly when we look at the activity of the microbiome and the amount of short-chain fatty acids that they can produce. And these changes are strongly associated with indicators of gut health, right? So remember what I mentioned about bifidobacteria, for example. The benefit, however, during disease challenge in the pig model is still unclear. And that doesn't mean it's not there. It means we need to study it and prove that it exists or not. We also need to balance prebiotic activity with whole body energy metabolism. And in the human model, that may end up with a different optimum than in swine production. And then the final one, two implications I have, is that the diet ingredient composition has an important role to play in determining how much prebiotic activity is there in a control diet. So when you, for example, test a feed additive as a prebiotic, you should also test the prebiotic activity of the diet. Because as a nutritionist, I can formulate a diet with very low prebiotic activity, and now the feed additive works wonders. And then I can also design a diet that has very high prebiotic activity, and then the prebiotic activity of a feed additive might be less. And then the final one is, dietary fiber and starch, so carbohydrates, play an important role in the management of post-weaning diarrhea. I'd like to acknowledge the students and postdocs and research associates that did the work, my collaborators that worked on the surgical models and on the starches. And then I'd like to acknowledge the funding that came from national Canadian organizations such as Swine Innovation Pork, NSERC, Agriculture Canada, provincial organizations, and then the industry partners. Without them, many of these national or provincial dollars wouldn't flow. And with that, I'd like to conclude my presentation. Thank you very much.